I want this to be a little interactive. So if you have any questions or get stuck, we can discuss. And if there's too much of discussions, Sylvia has to help us to make breakout rooms or whatever. We will see.
Yes.
What happens. So I will start by sharing my screen and show you what I thought about. All right. So in my view, the best way to get to know things is to get your hands dirty, to try it out a little, to get a feeling for it, get an intuition. That really a good complement to the theory that you can read about another time. I thought that we should start with not deconvolution, but convolution, the opposite. And I prepared a few small images for you. There's a link on Canvas. And your first task will be to convolve them.
Okay, so now all this is in the Canvas Module 8 lecture introduction to deconvolution.
Right, it should look something like that. There's a link, or you can just see it here.
So we're downloading the workshop, right, or something like this.
Yeah, in the Canvas you find a link which goes to GitHub where there are 3 files. You have to click them individually and then use the download button, and you will get them. So I'll give you maybe.
So sorry, I'm. Is it images for the workshop? That's where we should click, right? Okay, images for the workshop, right?
Yes.
That's it. Then you do that, and then it opens. It goes to GitHub. Yes. Please, anyone, if you have issue following, you let us know. So what do you want us to do right now? Just
download images, and when you're done, you can.
Sorry, I have a question. Should we click on the green button written code and then download zip?
I can just briefly show you, I guess, how it should look like. So if you go follow the link, it will look like this. So there will be, so there are 4 images here. That's the TIFF files. So you click them first, click it, and then you press, now it's in here, but then you press this button to download.
Okay, thanks.
Okay. Let's see. Yeah, I'm sorry. You have to take the slightly off road. I didn't know exactly how to upload it to Canvas. I was in last minute.
Has everybody downloaded the 4 files?
I don't know if it's just me, but I don't see the download button. It just takes.
So if you click on the name of the file, you go to a different page, and on the right side you have a little arrow.
Yeah, I don't have the arrow.
The arrow down.
Really?
I don't have that. I don't know.
Okay, what I will do anyway is that I'm going to put the 4 images.
Or maybe it's possible to share them here in the chat, maybe? Is that possible?
Let me see.
To put them in the meeting chat.
I guess another way is that directly on the main page, press the green button code and download the zip. It will download all the files there, right?
Yes, you can click on the code and then.
Yeah, there's a download zip. You will get all the files.
Yes, that's correct.
Yeah.
So whoever, was it Lauren who had the problem? Did you all manage?
No, it was me. It was Lisa.
How is it now?
I'm trying with the code button, but I will try again.
Okay, I put them in the chat. also maybe that will work.
So is everybody okay now, or are we still having some issues with the download? Everyone's okay? Please let us know if you are not okay. Okay, I guess that means everybody's okay.
Okay, we go on. So you have those two images. Then I thought you would convol them in Fiji. So you would open Fiji, then open the images in Fiji, and then go to plugins and Deconvolution Lab 2, and then it's called Deconvolution Lab 2 Lab again, which will open this window here to the left. And then you can load the, if you already have the images open in Fiji, you press this button, which doesn't even have a label here on my computer, and then you choose get platform images from the platform, and then you can pick the open images. So for image, you will use this 2 dots, and for PSF4. And then under algorithm, you choose convolution noiseless. Then hit the run button.
Okay, how are you guys doing?
It works.
Good.
So works for one person. We'll give it a few seconds more, then I can go on and also show what you should get. Just to
Sorry, I have a
select
question.
Yep.
So the plugin, it's under plugin.
Yes.
I've opened the images, but under plugin, I do not see the deconvolution.
Oh, I thought it's always included in Fiji. Are you sure you open Fiji and not image J?
No, I open Fiji.
You open Fiji. And then you have a missing plugin. Let's see.
Am I the only one in this situation?
Mine doesn't work either, but I don't know.
You don't
you
see
don't have it.
the deconvolution lab, or?
I actually just downloaded because I downloaded in a different computer. So I didn't have it on my laptop. I went and downloaded and restarted Fiji, but it seems to have some Java errors or something like that. I need to check it out. What's wrong?
Okay. Is everybody seeing the deconvolution plugin?
I can see it.
Yeah.
Okay, then it's only me not seeing the deconvolution lab plugin, or?
Alright.
Then I will manage. So has everybody managed to run the stuff?
Yes.
Yeah,
Yeah? Okay.
just to make sure, was it so that we were convolving actually, not deconvolving right now?
Yes, that's right. We start that way.
Okay.
So just keep it going. I will download the plugin.
Yep.
Why mine is not
Right.
there.
All right. So this is how it starts then. So it simulates actually what would happen if you image something that is super tiny and there's a point spread function that spreads it out. Now in this case it's just a Gaussian function, so it looks similar to STED maybe. And what you would see is these two bigger balls, or the 2 point spread functions, so one overlapping each input point. and they are just about resolvable. You can still see them as two. Right. So if deconvolution works, you would be able to use the point spread function in the middle and this result to get back to the inputs. That will be the purpose of deconvolution. So to go the other way, if you would save the result to a separate file, or otherwise it would just be called result, but it might be a good idea to save it if there's a mistake and you want to go back. And then for algorithm, instead you will pick, instead of convolution, you will pick Richardson-Lucy. And it will suggest, I think, 25 iterations by default. So that will be the next step. See if you can get back the input image again. 2 dots. Are you catching up, Sylvie?
Yeah.
Right.
Yeah, I get it back, but it's not exactly the same. It doesn't look exactly the same 2 dots.
Yeah, I have the same. It doesn't look so sharp.
Right. That is the expected result. So that's all right. Anyone wants us to wait a little more before continuing? Or you got there? I'll continue. So this is what I got. So I get 2 dots. They're a little smeared out, so it's not these tiny points. And why is that? So if you read about this method, you will figure out it's iterative. It does a little at a time. And the default 25 iterations, that's not enough to get back to the input. And actually it converges very slow, so you will not get numerically exactly the inputs, regardless of the number of iterations, probably. But now that you have it set up, I thought of 4 things you could try if you find any of them interesting. And yeah, so the first question is, how many iterations do you need before this convolved image looks identical to the input image again? So you could try a little more. Now I picked a very tiny 2D image, so you should be able to do at least 1,000 iterations even on a laptop, I think. And the other question is, so number 2, if you deconvolve this image you generated by the wrong PSF, so a PSF that is smaller or larger, what happens in that case? And you could also do 0.2, but you could start by creating a new image based on the larger PSF, PSF 8. And as the fourth alternative, you could start over and convolve with noise also. So then you would pick another algorithm. So it's a simulation with noise. So you could start that way also and see if you get back to the 2 dots.
Sorry, can I ask you about the algorithm that we used for the first image to make convolve it?
Yes.
What was the name of the algorithm again?
So convolution is a standard method that you find in mathematics. So it's just called convolution, and typically it's done in the Fourier domain. Something you will talk about later today, maybe.
Yeah, tomorrow.
Oh, tomorrow. Okay, maybe not for this purpose. But the idea is simply that in this case, if you have a image with just 2 points in the PSF, it will place the PSF on top of each of these bright pixels. But if you want a longer explanation, I recommend just go to Wikipedia and read about convolution. And in general, convolution is very simple compared to deconvolution. So it's really a simple process for a computer. It would just take this template of PSF and place it wherever you want with some intensity. So if you pick one of these, I will show you what you would expect to get for all cases a little later. So pick whatever you think is interesting, and we will hold here for a few minutes, I guess, while you do it. And any question, just feel free to talk.
So actually, I have a question, because at the beginning, when you were taking like get platform image from the platform, is there which image were you using? Is it the
So-
PFS4
So in the beginning,
at the start? Yeah.
yeah, yes, PSF4.
PFS. Oh, okay.
So the middle size, you will see it's the sigma of the Gaussian, actually.
And then you just say run.
Yes.
Mine says not valid, but so all I did, I now finally got the plugin, and so I open the Deconvolve lab, and then I say Deconvolve lab to lab or run, or which one should I say?
The one that says lab.
Lab, exactly. And then I get this window that looks the same as the one you have on your stuff. And there I select on the choose at the beginning, I select the PFS4 image.
Right. And it says so, and you still have it open. If you choose a platform image, it has to be open, I guess.
Sorry. So I'm opening the plugin, and then I just load the PFS4. That's all I do. And then I click run, or I need to do something.
Also the 2 dots.
Sorry,
image.
that one I didn't see.
Right. So it has
Yeah,
to be.
okay, right. Convolution noiseless. Yes, now I understand. Sorry.
Sorry, can you show us the 4 different aims again?
Right.
Thank you.
Eric, the algorithm for a convolution with noise is one that is called simulation with noise?
Yes, it's like in this image here to the bottom right. And yeah, and you can say how severe you want it.
Okay.
I did it, I visualizer just with the Poissonian, but if you use anything else, it will look worse than what I will show you. Okay, I'll continue in 30 seconds. So it doesn't matter if you don't finish any of them. All right, I intend to go on. So I have to. So let's see. We start with the first question. If we increase the number of iterations, what happens? And so the first issue was 25. If we take 5 times that, 125, you can still see a small pattern around the dots that look like small crosses. If you take it 5 times more, up to 625, visually it looks identical. But if you play with the contrast or use the other function in Fiji to compare images, you will see that it's not identical to the input, still not identical. And I would say in general, quite many iterations are needed with this vanilla Richardson-Lucy. There are acceleration techniques that use less iterations to get to the same results, but they require a little more memory. But that is probably what you would use, or something else if you use a commercial program. I don't know exactly what they're using. But most of them are based on Richardson-Lucy in some way, and many iterations might be needed. The danger is that if you have very noisy data, there could be artifacts also by doing this. Richardson-Lucy is known to amplify shot noise. So be a little careful and always double check with your raw data to see if it makes sense, because at some point it will start to look worse.
So I have a question about this. Is there any advantage in denoising the image first?
There is, and there was a paper in Nature last year about that. I think even I saw it in Hans' lecture. So there's a paper called it's called sparse deconvolution, I think.
Yes, you spoke about that. Yeah.
And if you go through this supplementary, you will see that it's actually a noise filtering step before using Richardson-Lucy, or alternatively the Landweber model if it's not that noisy. So there is an advantage, but it makes things a little trickier to interpret. And of course, there's a great benefit if you correct for problems with if there's a CCD detector. You have to characterize each pixel, or if it's CMOS, you will go over and correct for if there's a bias in the columns, for example. So those things should be done before deconvolution. Right. The other question is if you deconvolve it using the wrong PSF. So we created an image in the middle with a PSF4, and here it's deconvolved again, just 25 iterations. If you use the smaller PSF, it will never converge back. It will always look a little blurry. If you use a larger PSF, it might be that you get a bar at the end or something like that. And now this is the simplest PSF you can use. So imagine using something more complicated with a diffraction pattern, you will maybe appreciate the dangers, or we need to have a PSF model that matches what you have in the scope. So inspect the data, look at beats, see how it looks like, and make sure that the PSF you use matches what you see in the scope.
So it means you would never recommend using an artificial PSF?
Depends. So for fish dots, RNA or DNA fish, the artificial work very well because most of interest is quite local. But if you want to deconvolve, for example, nuclei or even thicker sample, more complex things, then maybe wide field isn't the right technique to start with because all samples are optically active. So the PSF will vary across the image depending on where you are.
I see.
But in most cases, so I work mostly with RNA and DNA fish, and there we use the theoretical ones.
So then I have the same question that I asked Hans then. Do you have any issues with people not putting their sample on the cover slip, putting it on the slide, and having a thick coat of mounting medium, and that distorts the PSF from one sample to the next in a different way?
That is more than I know. I don't know. To be honest, I never used the microscope. Besides, of course, I wrote software to.
So you're getting the images basically, and then okay, I understand.
And the latest problems we had with our scope was that there was a sample holder that was loose and blocked part of the light, so all images were dim on one side. So there's always something that can be problematic.
So that would be very interesting. Maybe we should have a look and figure out and take 3 samples and on the slide and compare them after deconvolution. Maybe we will really understand. I think that would be pretty cool.
Yeah. And another thing you can do, if you deconvolve, then you can convolve again with the PSF and see how different it is. The other thing here was that you could use the biggest PSF, PSF8. And if you use that for convolution, what you get is something that is not really resolved anymore. It looks like just from a big blob. So it doesn't meet the Braille criterion anymore. But even in this case, it can be deconvolved into separate dots. But of course, this case really requires that the PSF really matches. And here we are cheating because there is no noise, and we use the same PSF for both for generating the synthetic wide field or STED or whatever it is, and for deconvolution later on. The deconvolution can surpass the resolution limit in some cases. If the data quality is really high, it can, but there are also some ambiguities here. So if we would imagine not just 2 points, but imagine a circle of points instead. If it would look completely homogeneous, deconvolution, there would be different ways to place the underlying signals. So you could generate the same signal in different ways. What I'm trying to say while I stumble is that sometimes there's not a unique solution. There can be different solutions. And the last thing, point 4, was what happens if you use noise. And in this case, I used a little Poissonian noise, so it didn't affect that much. It made the signals look irregular, but it still could resolve it into 2 separate points. So this pitches on Lucy method is great for Poissonian noise, but it doesn't like the Gaussian noise that's typical from the sensor. It's sensitive to that. So if you redo this at home with more noise, you will see that crazy things happens at some point. Right. We still have a few more minutes, so I wanted to show you a few things that I think you should be aware of. One thing is what happens at boundaries of the image. Here I took an image from EPFL, which you also can download. It's a C. elegans embryo. And if you deconvolve it with the PSF that they supply, you get an image like to the right. It looks quite all right. And here, this image is kind of typical to most demonstration images that you find on the web, because whatever there is, there's it's black all around the thing of interest, not just in the lateral plane, but also actually you will see that there is something black on top and on the bottom of the object. But what happens if you image, you will, of course, always at some point, you will image and you will have a nuclei or something at the edge of the image. And if you use the this implementation in Deconvolution Lab 2, you will find if you deconvolve it, it will look black on the sides. So, meaning that if you deconvolve and crop, it will look fine on the edges, but if you crop and then deconvolve, there will be problems at the edges. And it's a little technical why it is like that, but it's related to the Fourier transform again, how these things are implemented.
So does this count? Do you think this is an artifact or a sign or something, or is just consequences of us deconvolving?
This is a consequence of using the Fourier transform without any padding. So what actually happens is that the algorithm it sees that the top and the bottom are connected. So what you see is that the dark thing at the top it merges to the bottom also. So it kind of wraps around at the edges. So left and right is the same. So you can imagine it's like a it's rolled up. Top and bottom are connected. Left and right are connected. So whatever is on the right will spill over to the left and so on.
And this is like some algorithms are made that way.
Yeah, they are implemented that way. Typically, the Richardson-Lucy algorithm doesn't really specify how you should do it. So the standard way is to use some padding, and there are different techniques to do that. There are a few options in Deconvolution Lab 2 to do that. Another example I have on this is if you have a full C-stack, which is not padded. So you see that the nuclei is the imaging started just on top and it ended a little before it you hit the bottom of the nuclei.
So when you're saying padded, you mean that we should start the Z-stack before the object, well before, and well after, and stop well after. Is that what you mean?
That's one way to do it. Yes, that's I think the standard procedure.
Okay.
If you don't, you will end up with something like this. For example, in Deconvolution Lab 2, if you use the default settings, that you will see that the nuclei will be just a thin line in the middle. It looks very weird. If you use the padding option in Deconvolution Lab 2, and that will be a longer lecture, you can prevent some of this. It's still not perfect because you see it's a little brighter at the bottom. If you would use even less slices, so here the nucleus is like you only imaged half of the nuclei, it will look even worse. And I'm sorry, the scaling here is not really perfect, but you will always see this bright thing at the bottom unless you have really unless there's some slack on top and on the bottom.
So you recommend that if we acquire a Z-stack to then deconvolve, we should acquire a Z-stack that is quite a lot larger than the object to make sure we have all the PSF information above the object and below the object so that we get maybe less of this issue.
So it depends on what software that you're using. If you're using Deconvolution Lab 2, that's the best way to go ahead.
Okay.
You should also read about their padding options. But yes, that's what I recommend. Be a little careful around the edges, not just in the lateral plane, but also in the axial direction. Mm-hmm. Which brings me to the point where you work. Because you have this portfolio assignment number 8. deconvolve your own images. And I will just, so obviously you will not have time now before the next lecture, but just to give you some clues, what you need to do in this case, if you have wide field images, it will be simplest because then we'll use the PSF generator, which also included in Fiji. And what you need to do is just open this PSF generator and fill in the details. So if you have oil, air, or water, the emission wavelength, the pixel size, so in the images, numerical aperture, and the distance between the planes. And then you also need to specify the size of the point spread function. And if you do without any padding, you should have at least as many Z planes, or probably you should match the number of Z planes with the number of Z planes in your image.
And the XY, size XY, is it the size of the image?
It will be the size of the point spread function. And what you can do, if you can generate a PSF, and you will see if you open it, you go to the top and bottom plane, you will see if the circular pattern is cut off or not. Ideally, you should see the whole circle in the top or in the bottom there. And also, of course, get to know your microscope. If you can, look at fluorescent beads, and always test on smaller images to figure out the settings. Because it takes some time. So use as small image as possible, and ideally crop out something that it has kind of nothing around. So if you have isolated nuclei, you can cut it out and just try on that one to see that you get the parameters correct, and then you can try on the whole image. Especially if you want to figure out the number of iterations, test on small data, and then you can run it overnight with everything. And yeah, I think you will see later on in the lecture from Nikon that there are other options also, but there are for sure faster programs than the Deconvolution Lab at this point. But this is very good for learning. I think still it's superb. It's just that no one would use it for high throughput data anymore. So with that, I think we have a few minutes for questions, and I wish you good luck with this assignment and the rest of the course.
Thank you very much. So, has everybody managed to do a couple of tests in this? So you're going to have this assignment where you have to try the PSF plugin yourself with your own images. And then you will need to tell us what happened. Yes, Yufei?
I'm not clear about the PSF generator because I don't have the PSF feed. And what's this step?
What the C step is?
Yeah, I don't have this information in my image now.
Because it's not a Z stack, you mean?
Yeah.
Okay, so you only have one plane. Okay, in that case, it doesn't matter, because the what you would do is you will set the in the size, you will set the Z value to 1, and then you will get just a single plane.
Okay.
So this C step, it doesn't apply if it's just one plane, if it's 2D.
Yeah, so I think in the assignment, the portfolio assignment that you have, there is two algorithms or two plugins that you have to try. One is called 2D deconvolution, the other one 3D deconvolution, and you have little videos that show you how to use them. It's quite straightforward, so I think it should be okay. Otherwise, you can ask us. Aphrodite.
Yes, I don't understand this size XYZ. Where will we find this information?
The pixel, the point spread function.
Yes, so it's not information that you get. It's something you have to just put yourself. So it tells you how big the PSF should be that this program simulates.
So it's the result, this number.
Yes.
Yeah,
So you can just use the default. It should be okay in most cases.
Okay.
And if you for some reason want to increase it, then you can do it. But you should be able to get good images with this in most cases.
Okay.
It's the default is set high. So actually, if you know what you're doing, you can probably use a lower number. It will be a little quicker, but it doesn't matter much.
Okay. Thank you.
Okay, Eric, I want to thank you very much. That was very nice. So I'm looking forward to seeing the